Hi everyone, and welcome to our lecture, which is the introductory lecture on what exactly is microbiology and a little bit of the microbial world and you. And in this, we'll kind of review some of the general or basic biology and chemistry that you should have encountered somewhere along the way by now, and just to kind of refresh your memory. But if any of it, let's say you don't recall or you may not have encountered, simply just send me an email or a remind message and I could kind of give you a little more information if I don't give enough in today's lecture, since it is kind of meant to be a review. Okay? So the first thing you want to ask yourself in this class is, what exactly is microbiology? So think about it for a second. How would you define microbiology? Okay. Think about breaking the word down. Micro and biology. So when you see biology, you know that that's the study of living things. And when you hear the word micro with something, what does that then tell you? Well, they're really small, right? So in this class, we are going to study living things that are very small, organisms that are not visible to the naked eye. So I want you to think about, which organisms would that be? Well, usually there are three that we focus on: bacteria, fungi, and viruses. Now you may want to ask yourself, why would we want to study bacteria, fungi, viruses, any of that sort of thing? What's the benefit of studying them? Well, think about it. There are a few different benefits. First off is the obvious one, especially if any of you plan on going into nursing or the medical field. Well, the first thing that we can learn by studying these things is how to understand them and their processes, so that we can determine how to destroy them or how to hinder them when they are causing problems to humans. Meaning, how do we treat humans who are being infected by the pathogenic ones of these microbes? Okay, so that's a big reason for studying microbiology, is the medical aspect, the understanding of these microbes to better treat patients. Another reason to study microbiology is the drug or antibiotic development aspect. Okay, so by studying these pathogens or potential pathogens, we're able to develop different drugs or antibiotics to help treat them. My personal favorite reason for studying microbiology is food production. So in the next slide, we'll kind of mention this a little bit too. Think about how many foods and drink products you wouldn't have if we never studied microbes or their processes, such as fermentation, right? Think about things like yogurt, cheeses, breads, beers, alcohol, any of that we wouldn't have without understanding microbiology. Lastly, microbiology is important for industry aspects. Okay, so the research field and obtaining different enzymes and various reagents. And we'll kind of touch upon these things throughout the course. Now, as I was just hinting to a minute ago, not all microbes are actually pathogenic or a problem. In fact, less than 5% of the microbes in the world are actually pathogenic. Okay? Most microbes either have no effect on humans or can actually benefit humans. So before we spend the course looking at various pathogenic or bad guy microbes, I want you to take a minute to kind of think about, what are some beneficial microbes? Okay? How are some microbes actually helpful to humans? Now, we touched upon some of these a minute ago. For instance, food-based microbes. So being able to have things like yogurt, probiotics, right? In addition to that, one of the things we're going to keep mentioning throughout this course is your normal flora. There are tons of microbes covering the outside and inside of your body that actually help you every day. So one of the most famous categories of normal flora are your gut normal flora, right? If you've ever heard of all of those bacteria living in your digestive tract, well, you wouldn't be able to digest certain foods, including a lot of vegetable-based foods with cellulose in them, if you didn't have those bacteria, those microbes, using their enzymes to help you digest your food. Normal flora is also very important because think about it. If all of these bacteria are coating your body, then they're preventing the bad guys from being able to have access. So to continue on that thought, let's go to the next slide now. The idea of good versus bad microbes. So like I just mentioned, we all have normal flora, and I want you to circle this word on your slides because this is very important. It's going to keep coming back to you. Normal flora are all of the microbes on or in your body that are not associated with disease. Okay, not associated with disease at that moment. Okay, these are the microbes such as your gut normal flora that we were just talking about that are constantly living on or in your body and can actually help you, okay, by preventing other microbes from being able to live on your body. Now, the one downside about normal flora is that they can be what we call opportunistic pathogens. Okay, opportunistic pathogens, underline this word. Okay, this will keep coming back in this course. Opportunistic pathogens mean that even though normal flora are normally not bad guys, they are normally not pathogenic, under certain circumstances they can become pathogens. Okay, I want you to think about what kind of circumstances might make these good guys that are living on your body, just chilling on your skin or in your gut, what might suddenly make them a problem for you and give you symptoms. Okay, well, there are two main times when that will happen, when normal flora become opportunistic pathogens. The first one is if you ever become immunocompromised, right? So you're in a weakened state and these guys are opportunistic. So what are they going to do? They're going to jump on that chance, right? They're going to take advantage of your weakened state and now they will be pathogenic and hurt you. The other time that normal flora become opportunistic pathogens is if they end up in the wrong place. So for instance, if those gut normal flora like E. coli that we were just mentioning a minute ago, if they suddenly end up somewhere other than your gut, for instance, your urinary tract, well, boom, now you have an infection. You get a urinary tract infection because these microbes are not supposed to be there normally, and so now they are considered pathogens. Okay, so whenever you see opportunistic pathogens, remember that normal flora can become opportunistic pathogens if you are 1, immunocompromised, 2, if they end up in the wrong place. So the way that I usually like to kind of give the analogy for students to remember this concept is, well, you may be the nicest, best, genuinely good student in the world, right? Normally not a thief or a cheater. But if, let's say, you were sitting in the professor's office, they happen to have the final exam sitting in your line of sight, and well, suddenly some other student came along, right? Emergency came up, the professor had to quickly leave the room and forgot that that final exam was right in front of you. You're not normally a cheater, right? But I'm pretty sure that if you were suddenly left alone for a few seconds with the final exam just right there in front of you, there's a good chance that your cell phone's going to come out and you're going to snap a few quick pictures, right? Even if you're the most honest person in the world. And it's understandable because it's opportunistic, right? The opportunity came up and the temptation arose, okay? And that's what happens to the normal flora as well. Now, once normal flora or any microbe causes disease, that's when we call it a pathogen, okay? And there are certain terms associated with this idea of microbes causing disease. The first one is pathogenicity, okay? Pathogen being a harmful microbe, right? Pathogenicity then means the potential or the ability for a microbe to now cause disease, to be harmful, okay? So pathogenicity, what I would do if I were you, I'd underline that P, that first P letter in pathogenicity, and then underline the word potential. So pathogenicity is the potential to cause disease. Whereas virulence is after that. Virulence is the degree of pathogenicity. How severe of damage can that microbe cause? Okay, so pathogenicity is the potential to cause disease, whereas virulence is how severe of a disease will be caused. Okay, now the biggest culprits of diseases in humans tend to be nosocomial infections. Anyone who's already kind of experienced any clinical work probably have heard this term. Nosocomial infection simply means any infection picked up in healthcare facilities, such as in the hospital. Now, unfortunately, one of the themes that we're going to keep seeing in microbiology is that over 250,000 Americans a year die from sepsis, okay? Meaning that over 250,000 Americans each year die because they picked up an infection in the hospital, and sometimes that's going to be from those of you who are working in the medical field. Okay, so throughout this course, really think about how the stuff that we learn can help protect you, help protect future patients if you're going into the medical field, or even just help protect your loved ones if you're not going into the medical field, because everyone knows that at some point or another, you or your loved ones will have to walk into a hospital or a clinic or doctor's office, okay? So keep these things in mind that we go throughout the course. Now, if one of these microbes does become a bad guy, become pathogenic, it must be able to do 5 steps. And again, this is something that you'll see come back throughout the course. So make sure the next slide that we turn to, you circle, star, highlight. Okay, so like I just said, put stars all over this slide. It will keep coming back, okay? When we think of pathogenic microbes, in order to be a pathogen or a dangerous bad guy, a microbe must be able to do these 5 things. The first thing they have to be able to do is enter your body or your cells, okay? And think about it. What are some ways that they might enter your body? Well, breathing, right? You have to breathe all day. So any of those openings you're using to breathe, that's easy access. What else do you do all day? Eat and drink, right? Eating or drinking are easy access. Any cut that you get or opened wound, those are easy access, okay? Sexual contact, that's another common entry for pathogens. Okay, so throughout the course, we're going to go through some of the various ways that microbes enter. But once they enter, in order to make you sick, they have to be able to stay in. If you instantly sneeze or, cough them out, well, then they wouldn't be a problem, right? They have to be able to attach and adhere, stay in your body. Okay, so now they've entered, they stay in. The next thing they have to do is defeat your defenses. Okay, so highlighting here the word defenses. What does defenses mean? In your body, that's your immune system, right? They need to be able to defeat your immune system, which is trying to destroy them. Once they've done that, they have to cause some sort of damage, okay? Because if they're not causing damage or not causing problems for you, then they're not a pathogen, right? You wouldn't even notice them. The last thing they have to do to be a true pathogen is transmit to a new host, okay? Because think about it. If this little bacteria only made one person ever in all of history sick, who never transmitted to any other hosts, well, then we wouldn't really know about it, and we wouldn't really consider it a pathogen or a problem. Okay? So just remember, in order to be a pathogen, a microbe must enter, stay in, defeat your defenses, cause damage, and then transmit to a new host. Now, before we go into all the details of fun infections and medical implications and everything, we have to review a few basic concepts first. Okay, so today we'll review chemical principles, meaning basic chemistry, and we'll also review some information about metabolism as well. So the first thing to review when it comes to chemistry are atoms. And as you may already know, atoms are the smallest unit of an element, which still displays the properties of that element. And what's important about atoms are the 3 subatomic particles that make them up. Okay, so in this course, what I'm going to want you to be able to do is be able to determine how many protons, neutrons, and electrons an atom has based on some information I give you. Now, before we can do that, you just need to remind yourself, what do we actually mean by each of these terms? Okay, so the first one, protons. Well, when you see that P letter of protons, that's positive. Okay, protons are positive. Now, where do you find them? In the nucleus. Okay, in the nucleus of an atom. Then you have neutrons. And like that name suggests, these guys are neutral, meaning they don't have any charge. And these you find also in the nucleus with the protons. So whenever you draw out an atom, the first thing you want to do is draw the nucleus with the protons and the neutrons. You then have the electrons circling that nucleus, okay, in their electron shells. And electrons are negatively charged. And electrons are the subatomic particles that will be moving around in reactions. So when we talk about reactions, an atom is either going to be gaining or losing electrons. Okay, they will not be changing their protons or neutrons. Now, when we talk about the protons and neutrons being in the nucleus and then the electrons circling, there's certain rules that go about how the electrons are circling. Okay, there's much more complicated rules than the ones that I will go over, but for the concept or the purposes of microbiology, you just have to know the general Bohr model or octet rule, meaning that when you are mapping out electrons, the first electron shell can only hold up to 2 electrons. Then each of the outer shells can hold up to 8 electrons. So for instance, the one that I am circling right now, carbon, what do you notice about the outer shell? Well, it has 1, 2, 3, 4 electrons, which means that this atom, to be truly happy, would want 4 additional electrons. Okay, so that will guide how it interacts with other atoms and what reactions it will undergo. Now, when we talk about atoms interacting to make their outer shells happy, to have that proper number of electrons. The general idea is that if an atom has three or fewer electrons in their outer shell, they tend to donate their electrons. Whereas if they have 5 or more, they tend to receive electrons. Because think about it. If you had 3 or fewer electrons and you need 8 in the outer shell, well, then it's quite a bit of work to try and fill that outer shell. You have to find 5, 6, 7 even electrons to fill that outer shell. So it's easier for you to just get rid of the 1, 2, or 3 electrons that you currently have. Whereas if you already have 5 or 6 or 7 electrons even, all you need is a couple more electrons to be full, to be happy. Okay, so in that sense, you would then just want to receive or gain electrons. Now, when we look at these atoms, okay, like I said before, some of the information that you'll need to know with regard to these atoms is shown here in their little icons. Okay, I'll always write out the words, but this shows it here. So for any atom, you'll see an atomic mass, which is the bigger number, and an atomic number, which is the smaller number. And that information is what we're going to use to figure out how many electrons, protons, and neutrons an atom has. Okay. So first, just to kind of make sure that you're comfortable with the octet rule, I had written a question on your slides and you did not have the picture there, but now I drew out for you how we would draw an element having 9 electrons. Okay, so in this slide, all I want to do is focus on the electrons to first make sure that you're comfortable with the octet rule. After that, the next slide that we do is how you'll actually start to encounter things more in the exam kind of context, in that I'll also be interested in you knowing how many protons, how many neutrons exist. Okay, but first, let's just look at the electrons. So if an element has 9 electrons, how would you draw it? Well, notice that the first thing that I did was I drew the nucleus. We then had the first electron shell, which we said can only hold up to 2 electrons. So now we have 2 electrons, but we need 9. So now we need a second ring, okay? And I always like to place them 1, 2, 3, 4 first, so that I could then easily see where the other electrons need to be placed. So we have 1, 2. You then go 3, 4, 5, 6, 7, 8, 9. Now, what do you notice about this atom? Is it full? Is it happy? No, because notice the way that I drew out the outer electrons, right away you can see there's a gap here. This guy can still have 1 more electron. Then it would be happy and it would be full, right? So what would he want to do if he was going to go into a reaction, if he's missing only 1 electron? Would he want to get rid of all of these other electrons? No, that would be so much extra work. Instead, what he would want to do is accept 1 electron. Okay, so he would be receiving an electron from an atom that then donates. Okay. If you have any trouble in terms of figuring out how to draw electrons on an atom, just send me a message in Remind or on email, and I'll give you a little extra practice. Okay. So now, as I just mentioned, when you actually encounter this kind of a problem, more like the exam setting, it'll be a little more detailed than simply kind of figuring out the electrons. You also have to be able to determine how many protons and how many neutrons would be involved as well. So now when you encounter a question like this, the first thing you're going to want to do make your nucleus. So in the nucleus, okay, our question tells us that the atom has a -1 charge, an atomic number of 7, and an atomic mass of 15. So the first thing you're going to want to do is circle atomic number of 7 and make note that this is always the first thing you're going to want to work with when you have one of these problems. The atomic number tells you the number of protons. So when you see atomic number of 7, you want to write 7 protons in the nucleus. Then you see that you have an atomic mass of 15. As I wrote on the side, atomic mass is your protons plus your neutrons. So if we know that we have 7 protons already, and our total is 15 for atomic mass, all you do is find the difference between those two numbers. So 15 minus 7, that gives you 8 neutrons. Okay, 8 neutrons in the nucleus. The last thing to do is to try and figure out how many electrons are present. Now, if we had a neutral atom, then that would tell you that you have just as many positive protons as you have negative electrons. So they would cancel each other out. So that would be 7 and 7. But in this case, we don't have a neutral atom. What do we have? A -1 charge. Since that charge is minus, what do you know that is negative when it comes to subatomic particles? Well, that would be electrons. So if you have a -1 charge, that means you have 1 additional negative electron. So instead of having 7 that would make this all even, you have 8 electrons. So what you then do is you have your 1, 2 electrons in the first ring, then you have 3, 4, 5, 6, 7, 8 electrons here in total. So when you look at this picture, just like the last slide, now what do you notice? Well, this atom is missing 2 electrons to be truly happy. So what would this atom want to do? It would want to react with another atom that can give it 2 electrons. So this guy would want to receive electrons, okay? Because it's easier to receive 2 rather than trying to get rid of all of these electrons that it has around it, okay? So again, if you have any trouble with this kind of problem, simply send me a message on Remind or an email. Now that brings us to the next slide. In this slide, this is really the way that you would encounter exactly on an exam, okay, this type of problem. Now what I want you to do is I want you to read this question, which is if an atom has a +2 charge, an atomic number of 10, and an atomic mass of 14, then how would you answer the following questions? And notice I put them out of order. Even though normally the first thing you would want to find is protons, I purposely put them out of order, okay? Because on an exam, you would usually only receive one of the questions, and you don't know which one it would be, okay? So now what I want you to do is to figure out the answer to each of those 3 questions, and then send me your answers as a Remind message answer, okay? Through the Remind app or the Remind website, you can just simply send me each of those 3 answers, and I'll let you know if you're correct. That way you can practice towards the exam. Okay, so now time for a little bonding. I know it sounds very sweet, doesn't it? So this is what we were talking about a little while ago when we said that all of the reactions or actions of an atom is basically based upon how many electrons it has in its outer shell and what would make it happy. You know, how does it get that full outer shell? So there are a few different ways for these atoms to interact with each other. The three main categories of bonds are ionic bonds, covalent bonds, and hydrogen bonds. Now, as I just mentioned quite a few times, this is all based on the actions of electrons, okay, trying to get that happy, full outer shell. And what we call all of this is reactions as these atoms are interacting with each other. So we're going to go through each of these types of bonds one by one. The first type of bonding is ionic bonding. Okay, ionic bonding means the exchange of electrons. So one atom is donating electrons while the other atom is receiving electrons. Okay, now what you'll notice is once that has occurred, one of the atoms will now be what we call an anion, and the other will become a cation. So you have to know the difference between these two terms. And there's actually a trick that I always use to remember which is which. The first thing that I do is in anion, I underline that N and I write negative. So an anion is the atom that has become negatively charged. Well, how would I become negatively charged? That means that I gained electrons, right? Because electrons are negative. Whereas the other atom, that's a cation. So what I always do for that one is I underline cat and draw a cute little kitty cat, right? Because cats are positive things. Well, unless you're allergic. But they're cute, adorable, positive, right? So a cation is the atom that is now positive. And how would you be positive in this reaction? Well, you lost negative electrons. Okay, so whenever you see the terms anion and cation, remember that an anion is negatively charged because it gained electrons, whereas a cation is positively charged because it lost electrons. Okay, circle, star, highlight those two words because you will see them again. Now, we said this is a bond, meaning that the atoms are now kind of stuck together. And so the reason they stick together after giving up electrons and gaining electrons is we just said that one of those atoms is now positively charged, the other is negatively charged. And what do you know about opposites? Well, opposites attract. So now this positive stays close to the negative, because they are bonded with that opposites attract electronegativity, kind of keeping them close together. Okay, but make sure that you know the difference between anion and a cation. The next type of bonding is covalent bonding. And you have to ask yourself, how is covalent bonding different from ionic? Well, now, instead of these atoms completely giving up or gaining electrons, instead they are sharing. So in giant letters right on this slide, sharing electrons. Okay, this can be done as a single, double, or a triple bond. And the two terms that you'll have to know associated with this are nonpolar and polar bonds. Okay, so when you look at these, when you think about these kind of reactions, any covalent bond, they're currently sharing because one atom is not strong enough to completely take away or gain the electrons from the other, okay? But they may sometimes still have slight differences in their strength. So that brings us to the next slide, where we really look into what is meant by nonpolar and polar. So first up, we have nonpolar bonds. Okay, nonpolar means that both atoms are equal in strength. Okay, so neither one is pulling the electrons closer than the other. Whereas polar bonds, now one of the atoms is quite a bit stronger than the second one. So that means it's kind of like when you force a kid to share his toy when he doesn't really want to. It's like, okay, yeah, look, I'm sharing, I'll share, but I'm going to keep pulling it a little closer to me because I really, really want it and I'm the stronger, bigger kid. Okay? That's what polar bonds are. And I want you to put stars next to the fact that water is an example of a polar bond. This is very important because like dissolves like. Make sure you write that down. Like dissolves like, meaning that since water is polar, what can it dissolve? Anything else that is polar. Okay, so water will dissolve anything else that's polar. Those things that are polar, that it interacts with really well, we call hydrophilic. Okay, they're going to be hydrophilic, as opposed to nonpolar items that would be hydrophobic. Phobic, they want to stay away from water, right? So polar likes water, nonpolar does not like water. Okay, make sure you're clear on those two things. The last of the bonds that we'll talk about is hydrogen bonding. With hydrogen bonding, when you think of a water molecule, for instance, you have the partially positive hydrogen atoms, and then you have negative atoms such as oxygen or nitrogen, okay? And so there's that attraction again between opposites being attracted to each other. Now, when it comes to hydrogen bonding, the important thing to remember is that these kind of bonds are much weaker than the other bonds that we just talked about when they are individual, when it's just one hydrogen bond. But when you have a whole bunch of hydrogen bonds together, that can be very strong. So for instance, if you've ever seen a picture of a little bug walking on water, okay, he's not a magician. What he's able to do is walk on the network created by hydrogen bonds that are quite strong together. Now, the example of why this is so important that I want you to remember is DNA. Okay, we're going to go through DNA and how it replicates and everything in the next lecture. But what I want you to know right now is that DNA, you already have probably heard before, is a double helix, okay? 2 strands stuck together. The reason that they're stuck together is hydrogen bonds, okay? Now, that makes them stuck together pretty well because there's a whole bunch of hydrogen bonds at that moment. But since we just mentioned that hydrogen bonds are individually weak, what can then happen when cells need to replicate their DNA or express their DNA, they can then unzip the double strands, 1 hydrogen bond at a time. Okay, so this concept of hydrogen bonds being weak individually, strong together is critical for DNA because we have the ability to then unzip our DNA into single strands, to replicate it, and to express it, okay? Replicate it and express it. So make sure you write that down. Hydrogen bonding is important for DNA because it allows us to open up that DNA, unzip it into single strands for replication and for transcription, which we'll talk about more in the next lecture. Now, speaking of hydrogen bonding, that brings us to water. And there are a few different things that I want you to remember about water. Okay, water has a lot of very important qualities to it. The first aspect we've actually already mentioned, which is that water can dissolve things. Specifically, what does it dissolve? Well, we said like dissolves like. Okay, so water will dissolve any other polar substances. That makes it a solvent. Once it has dissolved substances in it, it is now a solution. And those dissolved substances within it are solutes. So the example that I usually give is if you picture putting salt into a water bottle. Okay, the salt is the solute. Okay, salt is the solute right here, this word here. When you then put it into the water, The water is a solvent, so it will dissolve that salt. And now that you have salt water, you have a solution. Okay, the solution is the salt water that you've created. Okay, that's one important property of water, that it's a solvent specifically for polar molecules. Another important quality of water is that it can build or break different bonds. Okay, it can build or break what we call polymers, okay, meaning substances that are made up of a whole bunch of monomers. Now, the two terms associated with this are dehydration synthesis and hydrolysis. So I always, anytime you say dehydration, I want you to quickly follow it up with synthesis. So you're always saying dehydration synthesis. Now, most of you probably already know what dehydration means, okay? If you've ever been dehydrated, that means you're lacking water, right? So dehydration means when you remove water, you have synthesis, okay? Meaning building. So the removal of water can build molecules, polymers, okay? Whereas hydrolysis, whenever you see lysis in biology, what does that mean? Lysis is breaking, bursting, okay? So hydrolysis means that when you add water to something, when you add water, now you're breaking bonds, okay? You're breaking apart those big polymers into their individual monomers. So for hydrolysis, I always have you picture a fire truck and picture the big fire hose spraying water to break down, let's say, a wall or a bunch of boxes even, okay? Breaks it down, that power of water to break things. So again, remember, dehydration synthesis, so removing water builds, whereas hydrolysis, adding water, breaks things down, okay? The next crucial property of water we've kind of already touched upon as well, the idea that because water is polar, right, you can have molecules, other molecules, be hydrophilic that are polar because they interact very well with water. Okay, so polar molecules are hydrophilic because they interact well with water. We already said like dissolves like, so anything polar gets dissolved by water. Whereas anything non-polar, they do not want to be anywhere near water, right? So they will be hydrophobic, afraid of the water, not dissolving, not interacting well with it at all. This is important because if you picture the inside of your body or inside of cells, everything will arrange itself based on whether it's hydrophobic and doesn't want to be near water, or whether it's hydrophilic and likes to be up against water, really snuggling in with that water. Okay? So a crucial property of water here is that since water is polar, it allows anything else polar to be hydrophilic and arrange itself next to water, whereas anything nonpolar will want to avoid water and be hydrophobic. Okay, and as I have here the bullet point of why is this concept significant, that's what I was just talking about with the arrangement of anything in your body or cells. So for instance, when you look at this figure here, this is your double layer plasma membrane. The components of this membrane arrange themselves so that anything hydrophilic will be touching water, and anything hydrophobic will hide itself to avoid water. Okay, so that's significant for structures arranging themselves. Okay. The next property that we have here, which is the last of the crucial properties of water, is actually my favorite. This is the high heat of vaporization of water. So what that means when you hear vaporization or vaporizing something, okay, you're turning it into a gas, right? So high heat of vaporization means that it takes a ton of energy to turn water into a gas, meaning to evaporate it, okay? Now, because it takes so much energy to do this, it means it's taking away heat when it does this, when water goes from liquid to gas. So now, in mathematical terms, to kind of explain what I mean by high heat of vaporization, when you go from 98 to 99 degrees Celsius with water, okay, that only takes away 1 calorie of energy. Okay, it takes 1 calorie in order to raise this 1 degree of water. Same thing when you're going from 99 to 100, only 1 calorie of energy to make that jump. Now, when you go from 100 to 101, meaning when you now want to turn that water into a gas and you have to break all of those hydrogen bonds that have been holding it together, boom, look at that. It's 540 times the amount of energy that we saw in the other examples here. Okay, so now to evaporate that water, it now takes 540 calories of heat energy, okay, to make that jump to evaporation. That's a whole lot of energy that's getting lost, that's getting taken away. Okay, so you're taking away all that heat. This is why sweating cools you. Okay, that's the significance of high heat of vaporization. When you sweat and you have that liquid all over your skin, now once it evaporates, it's taking all of that heat energy away from your skin with it. Because look at how much energy that took in this mathematical example. So that's why sweating cools, is because of the high heat of vaporization of water. Okay, so you may want to put little stars next to why sweating cools you off. It's the high heat of vaporization of water. Now this slide here is just to give you a little figure, a little diagram on what exactly is happening. And as you see, there is that massive jump in energy that it takes to vaporize the water. Okay? Now, that was all the background in terms of reactions and bonding and water. Now we want to go into more the biological side of things rather than the chemistry side of things. And what we have here are all of the biomolecules that you encounter in your body and in your cells. So you have 4 biomolecules, which are carbohydrates, lipids, proteins, and nucleic acids. And as we mentioned before, you have the polymers, which are the big molecules. And they're built up of a whole bunch of monomers. And remember, we said that adding water can break these things down. Removing water can synthesize or build them up. Okay? Now, when we go through each of these, you'll kind of want to think of what is the exact purpose or significance of each biomolecule. So the first biomolecule we have are carbohydrates. And whenever you think of carbs, what do you think of? Quick energy, right? You have your monosaccharides, disaccharides, polysaccharides, a whole bunch of sugars. And when you have sugar, you get, boom, quick burst of energy. So the main function of carbs is quick energy, okay? They also serve as a structural material. So things like cellulose, chitin, peptidoglycan, those are all examples of carbohydrates that help with structure. So for instance, when you look at the figure, this giant cactus maintains its rigid, solid structure because it has a whole lot of cellulose. This crab shell, or any crustacean shell, maintains a hard outer shell and structure because of chitin, which is a carbohydrate. And then the one you will encounter a whole bunch in this course and in lab is the bacterial cell wall maintains its structure because of peptidoglycan, which is a carbohydrate. So anytime you see any of these three words here, cellulose, chitin, or peptidoglycan, make sure to remember that they are carbohydrates, okay? All of those are carbohydrates for structure. And the other main function of carbs, we said, is quick energy. The next biomolecule we have is lipids. And when you hear lipids, what do you think of? Fats, right? And fats are a great source of stored energy. So for lipids, I want you to write down that the main function of lipids is stored energy. Lipids also serve the secondary function of protection, okay? So when you hear lipids, I want you to think stored long-term energy and protection, okay? And the examples of lipids would be things like fats, oils, waxes, steroids, or phospholipids that you find in your cell's plasma membranes. The next biomolecule are proteins. And when we talk about proteins, I usually say their function is everything. Okay, as you'll see from this list, there are a ton of different functions for proteins. Examples of proteins that are most important in this class are enzymes and antibodies. So put little stars next to antibodies and enzymes, and make sure you remember that both of these are examples of proteins. Other proteins that you might encounter are things like keratin and collagen for structure in your hair and nails. There are transport channels that are made of proteins, a lot of hormones for regulating different reactions in your body are made of proteins, and even the movement of your muscles is thanks to various different proteins. But like I said, the ones mainly encountered in our course of microbiology are enzymes and antibodies. Now, when it comes to proteins, one of the things you have to remember is that structure defines function, okay? Structure defines function, and there are 4 levels of structure or organization for proteins. Every protein will first of all have its primary structure, which is here. Primary structure is simply the chain of amino acids. Okay, so if I just wrote out a long chain of amino acids like lysine, alanine, glycine in a row, that would be the primary structure. What then happens is those amino acids, they have their R groups that we'll talk about in later lectures. And what this means is those amino acids will start interacting with each other and will form either an alpha helix or a beta sheet. Okay, so a single alpha helix or a single beta sheet, that is the secondary structure of a protein. When you then have multiple alpha helices interacting with multiple beta sheets, that now is the tertiary structure to form that globular shape, okay? When you then have multiple subunits making one giant polypeptide, that is quaternary structure. Okay, so remind yourself, primary structure, write down that is amino acid names. Secondary structure, write down single, one alpha helix or one beta sheet. A single alpha helix or single beta sheet is secondary, that S sound. Then write down that multiple alpha helices, multiple beta sheets, that is tertiary. And last, you have many different subunits together, that is quaternary. And you'll understand that more when we go over enzymes and various structures in micro. This slide is just to recap the various structures that we just went over. And again, next to secondary, you will want to write single for alpha helix or single beta sheet, whereas tertiary, you'll want to write multiple helices, multiple sheets, to really remember the difference between secondary structure and tertiary structure, because those are the two that I see students mix up the most out of these different structures. So as we were just saying, protein structure defines protein function. So if a protein loses that structure and does not have its proper primary, secondary, tertiary, quaternary structure, then it won't function properly, and it could cause major problems within your cells or your body. There are various diseases associated with it, some of which we'll even talk about in this course, such as mad cow disease, which you see on the list here, okay? What I really want you to know, though, from this slide is the idea of denaturing versus proteolysis. So both of these are a problem. In both cases of denaturing and proteolysis, the proteins do not have their proper structure anymore. The difference is, in denaturing, when a protein denatures, this does not destroy the primary structure. Okay, for denaturing, you can simply write unfolding protein. Okay, so for denaturing, write down that the protein has unfolded or misfolded. So it still has its primary structure, meaning the correct order of amino acids, but what it does not have anymore is secondary, tertiary, or quaternary structure. Proteolysis, on the other hand, when you see lysis in biology, what did we say that means? When you see lysis, that means breaking, bursting, destroying, right? So proteolysis, this has broken completely, chopped up the protein. So in proteolysis, you have now lost primary, secondary, tertiary, and quaternary structure. This is irreversible. Okay. Usually this is done by various enzymes that chop up a protein. But this is very, very detrimental, as it is irreversible and primary structure is lost. Okay, so remember, proteolysis, the primary structure is lost, chopped up protein. Whereas denaturing, that's an unfolded protein that still has its primary structure. The last of the biomolecules is my personal favorite. I'm known for loving DNA and RNA. Okay, with biomolecule number 4, we're looking at the nucleic acids. Nucleic acids are made up of nucleotides, and we're going to go through these much more detail in the next lecture when we cover genetics. Now, what this means is you have this structure of alternating sugar and nitrogen bases and phosphate groups, and you have them making RNA or DNA. They both have their own very specific structures and functions. Okay, when you see DNA, you have double helices, and DNA is known as the genetic material, as it then codes for RNA, and then RNA codes for protein. And that will be the central dogma that we learn in the next lecture. The central dogma of biology is DNA codes for RNA, which codes for protein. Now, when you look at DNA and RNA, I want you to think about the various differences between the two. Okay, you have DNA, which is deoxyribonucleic acid, which tells you that its sugar is deoxyribose, whereas RNA, the sugar is ribose. You then have DNA being double-stranded, whereas RNA is single-stranded. In DNA, one of the nitrogen bases that you have is thymine, or the letter T, whereas in RNA, you don't have the letter T. Instead, you have uracil, which is the letter U. The last difference is the one that we already mentioned, which is DNA codes for RNA, whereas RNA codes for protein. And what I'll do is on the next slide, I'll summarize these differences that I just rattled off here. So as we were just going over, this is just a recap of the differences that I told you between DNA and RNA. Okay, and like I mentioned, we'll go over more of this in detail in the next lecture. Just a few more things to mention as a brief review of nucleic acids before we get the details next time. The bases, the nitrogen bases of these guys are A, T, G, and C if it's DNA, A, U, G, and C if it's RNA. So that was on our list of differences. Notice that everything's the same here except for the T versus the U. The way that these match up, I'm going to skip ahead to over here, the complementary pairs. The way that these guys match up is A has the complement of T, G has the complement of C. So I like to think of it as at Georgian court, okay? At Georgian court, to remind you that whenever you're looking for a complementary base and you hear A, then the complement is T. If you hear G, the complement is C. And if you're looking to build RNA from DNA and you want to know what the complement of A is, well, it can't be T, right? Because RNA does not have T. So the complement of A when you're dealing with RNA is U, or uracil, okay? Now here you see just a little terminology, nucleoside versus nucleotide. I always underline T and write total. So nucleotide means the subunit of DNA which has the sugar, the phosphate, and the nitrogen base, whereas a nucleoside is only the nitrogen base and sugar. It does not include the phosphate group. Okay? The last thing to mention here with nucleic acids, and once again, this is something you'll see more in the next lecture, is that when you have DNA in a cell, DNA is crazy long, okay? Picture like 6 foot long worth of code, okay? In order to fit that in such a tiny cell, it has to be coiled in a way that it kind of is balled up, right? The way that bacteria do this is with gyrase or topoisomerase, okay? So I want you to circle this word here, gyrase or topoisomerase. I'll usually write it as just gyrase. Topoisomerase is a big fancy umbrella term, okay? Now what's important about gyrase is that the way that bacteria use gyrase to coil up their DNA, that's different from how our cells coil up our DNA. We humans do not have gyrase, okay? Meaning that as humans, we instead use what's called histone proteins. This is significant in the medical field because whenever you hear that bacteria has something that we do not, what can you then do? Target that, okay? You can target a bacteria's gyrase, kill the bacteria because it can't properly coil and uncoil its own DNA, but not hurt your patient because your patient doesn't have gyrase, so you're not hurting anything that they have. Okay, so with gyrase, it's important to remember that only bacteria have gyrase, and you'll see this come back in the next lecture. So make sure that you know that gyrase coils and uncoils DNA in bacteria. The last thing that I want to cover today is microbial metabolism. Okay. Whenever you hear the word metabolism, what metabolism means is all of the chemical processes in a cell or organism. Okay, so that means both building things up and breaking things down. I emphasize that it's both because a lot of students, when they hear metabolism, they just think of their own body and how people commonly use the phrase, oh, I have a fast metabolism. I'm really good at breaking that food down. But it's not just breaking things down. It is also building them up. So please make sure you circle or star or highlight the fact that metabolism is all of the chemical processes, building up and breaking down. Now, there are two terms associating with this for those chemical processes, and that is anabolic or catabolic reactions. Now, before you start thinking I'm a crazy cat lady, I am going to use another cat reference to help you visualize this. Whenever you're trying to think of the difference between anabolic reactions and catabolic reactions, when you see the word catabolic, I want you to picture a cat scratching apart a sofa, right? Just tearing it apart with their little claws. That is catabolic, right? That is a reaction in which you are breaking down large molecules into small molecules. So we have it in the figure here. Catabolic is breaking down large molecules into small molecules. So again, picture that cat tearing apart a sofa with its little claws. Then anabolic would be the opposite of that. Instead of breaking things down, in anabolic, you're building things up. Okay, so think about anabolic steroids, like think of A for Arnold Schwarzenegger, right? Anabolic steroids, building up muscle. Okay, so remember the difference between anabolic, building things up, versus catabolic, reactions that break things down. Now, any of these reactions can use what we call a catalyst, or in biology, an enzyme. Okay, enzyme and catalyst is the same thing, except that enzymes are specific to biological systems. Okay, these are simply substances that will help reactions, okay, help speed them up by lowering their activation energy. So I want you to circle that. Circle that they lower activation energy. That will be important. So speaking of why that's important, here we have enzyme action, right? How do enzymes help reactions? And as I just mentioned, enzymes help speed up, meaning quicken, reactions by lowering the activation energy. What this means is activation energy is the energy required to start up a reaction, right? To activate it. So if you lower that activation energy, that means that now that it's lower, you'll get to start up quicker, right? So it's quickening the start of that reaction. It's not getting used up. It's not changing the overall free energy, which would be the reactants versus the products. It's not changing any of that. All an enzyme is doing is helping you quick start that reaction. So kind of think of it as if you have been saving up money to buy a new cell phone, right? Currently costs $1,000. Let's say it's one of the fancy smartphones, full price, expensive. If you have to save up $1,000, that's going to take quite a bit of time. Whereas if they suddenly drop that price to $50, most of you can just go out and get it now. $50 is a lot easier to save up to and to get that ultimate goal versus having to wait and save up to $1,000. So that's what enzymes do. They drop that price. They make it quicker and easier to obtain this reaction. Okay, but they're not doing anything else. Okay, so free energy stays the same, but they lower the activation energy to grab that reaction. The analogy that I like to do with enzyme action, or how an enzyme actually works, the mechanism of enzymes. Let's look at the picture on the next page as we go through this analogy. Okay, so here you have an enzyme, right? This is an enzyme. It has active sites here for substrates. Okay? Substrates are simply the building blocks of a molecule. Okay? So for instance, amino acids that get put together or interact with each other to make proteins. So now you have this enzyme, and the analogy I like to make with the enzymes and their mechanism is enzymes are like a seat on a school bus. Okay? All it's doing is giving two people a place to sit next to each other, putting them in close proximity so that they can interact and start a reaction. So if you picture an enzyme like the seat on a school bus, well, what happens? First day of school, two little kids come in. They sit next to each other on this seat. What happens when you are near each other, especially little kids? What are they going to do? They're going to start chit-chatting, talking, interacting. By the end of that bus ride, they're like, oh my God, we're best friends, right? Because they have now bonded by sitting close to each other, by being in close proximity. Okay, that's exactly what enzymes do. They are a seat here for two substrates to sit next to each other in close proximity so that they will then interact and form a bond. And then what happens, just like the two little kids on that seat bus, they get up, they leave, right? They leave, and what happens, that seat is now available for two more kids to sit next to each other and interact and bond. And that's what happens with the enzyme. Once these substrates have bonded and leave, the enzyme has not been changed or used up in any way, and it allows for two other substrates to come along, sit on the enzyme, bond, interact, and then leave, okay? And through that process, if you think about it, by bringing two things in close proximity, okay, they're not actually part of the reaction, they're just facilitating it. They're allowing this reaction to happen quicker because it's forcing two substrates to sit near each other. Whereas if these substrates were, across the room, different parts of a building, they're not as likely to meet up with each other, to bond, okay? So that's what we mean in terms of lowering the activation energy. This enzyme, by having the two substrates right next to each other, they're quickening the start of the reaction. They're lowering the energy required for these two to bond. But again, they are not used up. They're not changing free energy, okay? Now, with this enzyme activity, there are various factors that can affect how well this enzyme is going to work, okay? How these reactions are going to go. Some of the factors include, first of all, substrate concentration. So ask yourself for each of these factors, how would enzyme activity be affected? Okay, make sure for each one you're able to briefly explain how this factor would affect enzyme activity. So first up, we have substrate concentration. If you have a lot more molecules, right? A lot more substrates present. What will that mean? More molecules or more substrates means more interactions. The substrates are more likely to bounce into each other and bond or interact. So you have increased activity, okay? More substrate increases activity, less substrate decreases activity. The next two factors are related to each other, optimal pH and optimal temperature. These two factors, if you think about it, what did we say enzymes are earlier in this lecture? Enzymes are proteins. What happens to proteins at the wrong pH or temperature? They denature, right? And when you hear denaturing, that means the function is ruined. So if an enzyme does not have the optimal pH or the optimal temperature, it will denature, and this reduces enzyme activity. Whereas optimal pH and optimal temperature allows for good enzyme activity. Then we have enzyme cofactors and coenzymes. For cofactors, I want you to circle factor and right above it, write the word factory. I want you to picture a factory, a big building and smoke and metals and minerals, all that stuff, because cofactors are metals or minerals. They are inorganic molecules that help activate enzymes, that help enzymes to work. Okay, so cofactors are inorganic metals or minerals that activate enzymes, whereas coenzymes, these are organic vitamins that activate enzymes. Okay, so one more time, cofactors are inorganic metals or minerals that activate enzymes, whereas coenzymes are organic vitamins that activate enzymes. Okay, so that's why if you think about even yourself, you always have to have your various minerals that you take in, things like zinc and magnesium, as well as coenzymes, things like vitamins, to help your cells have proper enzyme function. Another thing that can affect enzyme activity is enzyme inhibition. And there are 3 different ones that you have to know the difference between or amongst. Enzyme inhibition can be non-competitive. Okay, in non-competitive inhibition, which you see in this figure here, non-competitive because the inhibitor is not competing with the substrate for the active site. Notice the competitor is binding somewhere else on the enzyme, and that changes the shape of the enzyme, so the substrate can no longer fit. It can't sit down on this enzyme, so it's inhibited. But notice there's nothing actually blocking the active site itself. The inhibitor binds elsewhere. That's why it's non-competitive. It is not competing for the same site. Whereas competitive inhibition over here, you have the inhibitor physically blocking the active site. The inhibitor is competitively blocking the active site, so now the substrate cannot bind. Okay, so you do not get the reaction. The last type of inhibition is feedback inhibition. Okay, when you look at a body or cell, there's usually a whole bunch of reactions in one chain. Okay, so there's usually multiple enzymes involved to make one ultimate product. And many times this product then goes back and blocks one of the enzymes in that reaction. Because think about it, if you already have that product, why would you want to waste resources, time, energy making more of that product that you don't need? The cell would just fill up with that product and explode. Okay, so instead that product serves as an inhibitor to block enzymes in that pathway to block production of more product because you already have that product. Okay, so you have non-competitive, competitive, and feedback inhibition. Make sure that you're very clear on the differences amongst them. Now I want to point out one other thing is when you hear non-competitive inhibition and you hear a change in shape of an enzyme, you'll usually also hear the word allosteric. Okay, allosteric, A-L-L-O-S-T-E-R-I-C. Allosteric change simply means a change in the shape of the enzyme, and that you find in non-competitive inhibition. The last thing I want to go over with metabolic reactions and metabolism are the energy yielding reactions. Okay? When you look at this slide, there's a few different terms that you have to know. Okay? They're bolded, so you have chemoorganotrophs, chemolithotrophs, phototrophs, and these terms here, heterotrophs versus autotrophs. All of these terms I want you to be very comfortable with. The difference between these terms, first off, is what they are yielding or producing. Heterotrophs and autotrophs refer to organisms trying to obtain carbon. Okay, so heterotrophs and autotrophs is referring to what an organism is breaking down in order to obtain carbon. So heterotrophs means that an organism is breaking down organic matter to obtain carbon, whereas autotrophs means self-feeding. These organisms are breaking down inorganic matter, such as CO2 from the atmosphere, to obtain carbon. And what they do with that carbon is then build the biomolecules that we talked about. Okay, so make sure that you know heterotrophs means that an organism is breaking down organic material to get carbon, whereas autotrophs, they are breaking down inorganic material to get carbon. Notice both cases, they want carbon. These three terms, on the other hand, these three terms over here, they are breaking things down to get energy. Okay, not carbon, they're breaking things down to get energy. Chemoorganotrophs are breaking down organic molecules to get energy. Chemolithotrophs are breaking down inorganic molecules to get energy. And lastly, phototrophs are breaking down light to get energy. Okay, so notice these three are about energy. These two here are breaking things down to get carbon. Now, the last thing I want to point out on this slide is over here, you see organisms breaking down glucose to get energy. What do we call that? That's respiration or fermentation, right? That is breaking down glucose to get energy. Respiration, which we'll talk about in a minute. Now, in this case, since you have an organism breaking something down, would you consider that reaction catabolic or anabolic? And what you have to do is remember our little visual, right? Catabolic. Scratching, 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 scratching up that sofa, tearing it apart, breaking it down. So whenever you see breaking down glucose, well, that tells you that's catabolic. You are breaking something down. Okay? Once again, you may want to put little stars there and make sure that you know all 5 definitions that we discussed on this slide. Heterotrophs versus autotrophs, and then chemoorganotrophs versus chemolithotrophs versus phototrophs. Okay? Now, the last thing to go over in terms of metabolic processes are the examples of using organic material for energy, which are respiration versus fermentation. Now, if you go over respiration and fermentation in another class, such as biochemistry, you have to memorize all kinds of structures and enzymes and everything. For this course, you don't need that. What I want you to know for these 2 things is how they will differ in terms of presence or absence of oxygen, and how much ATP will an organism get in respiration versus fermentation, and whether they are a eukaryote or a prokaryote, which we'll talk about more in a couple of slides, but basically the idea is prokaryotes have no nucleus, no, okay, trick, no. Prokaryotes have no nucleus, whereas eukaryotes have a nucleus and internal membranes, and that will affect how much energy they get from this type of reaction. So the first thing that we have here is showing the difference between respiration versus fermentation. Notice that glycolysis is the first step in this breakdown of glucose or organic material. You can even cross out organic compound and just write glucose, because we're usually just talking about glucose here. And so notice that glycolysis occurs first, and it doesn't matter whether oxygen's present or not. Either way, glycolysis will occur, and either way, this step of glycolysis will produce 2 ATP, okay, 2 pieces of energy. Then what happens next depends on whether there's oxygen present. If there's no oxygen present, then the organism cannot undergo respiration and instead has to undergo fermentation. And what it does in fermentation is break down the products of glycolysis into lactic acid or ethanol. Okay, and that's why you hear if there's too much fermentation, your muscles, you're building up too much lactic acid. That's not good, right? Get fatigued with that. Now, with this fermentation, what do you notice? How does fermentation over here differ from glycolysis and from respiration? What are you not seeing over here? There is no ATP produced from the fermentation step. Okay, so if there's no oxygen present, you get no ATP from fermentation. So all you get from one glucose is 2 ATP if there is no oxygen present. Because remember, no matter what, glycolysis will happen. So you get 2 ATP no matter what, but then you get nothing extra from fermentation with regards to ATP. What you do get out of fermentation is that the products of fermentation can then go and serve as feeding into more glycolysis, which will give more ATP in that way. Okay? So now, that's if there's no oxygen present. If, however, there is oxygen present, a cell can undergo respiration. And with respiration, what do you notice? Boom, a whole lot of energy, a whole lot of ATP. In this case, up to 36 ATP from respiration. So if oxygen is present, how much ATP can that cell get from one glucose molecule in total? It could get up to 38 ATP because you have one glucose, no matter what, glycolysis occurs, so it gets 2 ATP there. And then if oxygen's present, it gets a whole lot more in respiration, up to 36. So that would be 38 total. So now we're going to look at how the textbooks or online would kind of break this down a little bit more. I kind of made this slide myself to just kind of show you the simple version because, like I said, in this course you don't need crazy details for micro with regard to this process. So let's look at a few more details before we finish up. So, continuing on with the metabolic process of breaking glucose down. We have 4 phases of respiration listed here if oxygen's present. Notice that glycolysis occurs whether oxygen is present or not, and that we call it anaerobic, okay? No oxygen needed. Whereas the rest of these processes here are aerobic. They require oxygen, okay? I want you to be familiar with each of these names of the processes involved in breaking a glucose down. You have glycolysis, prep reaction, citric acid cycle, and the ETC, okay? Electron transport chain coupled with chemiosmosis. You do not have to know the crazy mechanisms of each. You don't have to know pyruvate, acetyl-CoA, any of that. All I want you to know is these names associated with respiration. Know that glycolysis does not require oxygen. And note where you're getting your ATP from. Okay, so notice glycolysis gives 2 ATP. Citric acid gives 2 ATP. And electron transport chemiosmosis gives 34 ATP. What do you notice? One of these reactions does not give any ATP, and that's the prep reaction. Okay, so make sure to put a little star for yourself and write that the prep reaction is the only phase of respiration that does not give ATP, and it makes sense with its name. Think about if you're cooking, for instance. If you're prepping a meal, if you're cutting up a piece of chicken before cooking it, well, you can't eat it at that moment and get everything that you want from it. You're just prepping it. You can't get the energy from it until you actually cook it and eat it, okay? So prep reaction does not give any ATP. It simply prepares the molecules so that the cell can then break them down for proper ATP in the next steps, okay? So again, be aware, glycolysis, prep reaction, citric acid, and electron transport chain. Make note that glycolysis does not require oxygen, but the other three do. And make note that the prep reaction is the only step in this process that does not yield ATP. And the last thing to note here is how much total ATP can that cell get from one glucose. That total is 38 if oxygen is present. Now make note that that is if we are talking about a prokaryote, meaning something like bacteria. In a couple of slides, you'll get to see that a eukaryotic total is slightly different. Okay, so prokaryotes, if oxygen is present, can get 38 ATP. If oxygen is not present, then they must undergo fermentation instead. And as we mentioned in the last slide, fermentation does not give ATP by itself. Instead, there's only a 2 ATP yield from the glycolysis step. Okay, so make note that there's no oxygen present for fermentation and that the total yield when no oxygen present is just 2 ATP. Okay, and that will be the same whether it's a prokaryote or a eukaryote, because you'll notice that glycolysis takes place in a different spot than respiration in eukaryotes, which we'll talk about in a second. But again, keep in mind, oxygen versus no oxygen, how much ATP we are getting. This is just to show you an example of how you might encounter this process in a figure. Okay, again, this is more complicated than what you have to know. I simply put it here just to help you visualize respiration versus fermentation. But again, you don't have to know all the details in terms of NAD, NADH, any of that stuff. The last thing to note with metabolism is that difference that I kept hinting to with regards to prokaryotes versus eukaryotes. So like I said, the difference between a prokaryote and a eukaryote is that prokaryotes have no nucleus, they have no internal membranes. Okay? What that means is that all of these processes are able to occur in one location, which happens to be the same location where energy can be used in the cell. Everything's mixed together in a bacteria. Eukaryotes, on the other hand, we have internal membranes, right? We have nucleus, we have our various organelles. So in eukaryotes, when they make the ATP, they then have to pay a 2 ATP toll to get that energy across their internal membranes to where they'll actually use the energy. So what that means is eukaryotes will lose 2 ATP from their respiration process to get the energy where it needs to be. Now, what eukaryotes do not ever lose is 2 ATP that they gain from glycolysis, because glycolysis happens in their cytoplasm. There's no membranes to cross over. They get to keep that no matter which process they do. So as a recap, just to kind of go over the different amounts of ATP that cells can have depending on whether oxygen is present or not. If a cell is a prokaryote, if no oxygen is present, then from breaking down one glucose, they get 2 ATP, because remember, they get it from glycolysis, and then fermentation gives them no extra energy. In a prokaryote, if oxygen is present, they now get 38 ATP. They get the ATP from glycolysis. They then get it from their citric acid cycle, the electron transport chain as well. Okay? So they get 38, and they don't lose any because they don't have any toll to pay, no internal membranes. Eukaryotes, if oxygen is not present, they get 2 ATP because they get 2 ATP from glycolysis. And like I mentioned, glycolysis, there's no internal membranes there. Okay, it happens in their cytoplasm. So they get to keep that 2 ATP. If, however, oxygen is present in a eukaryote, then from one glucose, they would be able to produce 38 ATP, but they would only have a yield of 36 ATP from one glucose when oxygen's present, because they have to pay that toll. They have to use energy to get that ATP out of their internal organelles and into the cytoplasm where they want to use it. Okay? So I'll just write that out on the next slide to help you visualize each of those 4 scenarios that you have to know. So make sure to put little stars on them. Okay, so here is the recap that I promised you of how much energy each type of cell can yield from breaking down one glucose. So again, a prokaryote, no oxygen, they get 2 ATP. Oxygen, they get to keep 38 ATP. Eukaryotes, on the other hand, no oxygen, 2 ATP, just like a prokaryote would. But if oxygen is present, a eukaryote only gets to keep 36 ATP from breaking down one glucose. Okay, and again, that's because they have extra internal membranes. That's it for this lecture, and now it's time for you to head over to the discussion board where I'll explain an exercise that you'll do at the end of every lecture. Okay, basically I have you become the professor. It's a study technique that has helped a lot of my students really jump their grades from lower in the beginning of the semester to much higher at the end of the semester as they learn how to really focus in on what is significant to study. Sometimes students get caught up in being a student and in trying to memorize every little fact, rather than studying like a professor and just going slide by slide, asking themselves, if you were a professor, what are the questions that you could ask from this material, and how might you ask them? And that's basically what I'll have you do at the end of each lecture. You'll go into the discussion board and practice your study technique by each student posting a question that if they were the professor, they might ask from this lecture. Okay, each week we'll have a discussion board set up for each week's particular lecture. You can format it in short answer version. If there's, let's say, a multiple choice that you really want to ask, you could do that as well. The reason why I usually encourage short answer version is even though a lot of the questions you encounter on the actual exams will be multiple choice, if you study in the form of short answer questions, then you'll be very prepared to answer either multiple choice or essays that might encounter this topic. And it also helps you to better understand the concept as a whole. Okay, and ultimately this idea is that when you study like a professor, as you've probably already heard, the best way to really get to understand something is by teaching it to someone else. So if you start focusing in on how you would teach things to someone else or how you would ask questions, it kind of helps you in your studying. And basically keep in mind that when I make my exams, I make them from scratch. And I don't use any test banks or any textbook types of questions. Instead, I go through my slides that I gave you, and I go through what I said and what I wrote down, and I look at each slide and I do just like I'm asking you to do. I ask myself, what question can I ask from this slide? Okay? And if you're having any difficulty with any of the material we covered or with coming up with your question and then having to answer someone else's question on the discussion board as well, simply send me a message and remind or through email. Like I always say, I have no life. So I'm more than happy to answer any of your questions 24/7. Never feel like you are bothering me. Never feel like you ask too many questions. There is no such thing. I truly encourage and appreciate the questions, as it really shows me that you're taking this seriously and taking the time and dedication to go through this. Okay? Thank you for listening, and have a great day.